organized and deeply committed to advancing our research mission. Most of all, I want to thank today's panelists. I know we asked you to do a lot and to think lots of big thoughts, but the best part of it is we asked you to do that in a really short period of time. I think you really appreciated that part. It's been so impressive to hear both Ram and Megan talk about how you have worked together with your colleagues across grounds to develop the ideas we're going to hear about today. So, by way of background and just stepping back a little bit, President Ryan and I announced last summer, as part of the university strategic plan, we were committed to making substantial investments in brain and neuroscience, which is one of the 5 research areas staked out in the strategic plan as places where we want to move from prominence to preeminence, to really move the needle. Our goal in making these investments is that we want to increase our research capacity and its impact and help take our research to the next level. We wanted to focus on areas where we could bring together many different UVA strengths in STEM, in the social sciences, in the arts and humanities, and in translational and clinical settings to lead in these areas. We solicited input from across grounds on focus areas, and we assembled the interdisciplinary faculty panels you're going to see today. We gave those panels a big assignment. We asked them to come up with 1 or 2 moonshot ideas for each topic. I know that some people don't like the term moonshot, and prior to its use in this forum, I might have been one of them. It implies, I think, to some people that there's a single goal, getting to the moon. And obviously for brain and neuroscience, the problems are complex, they're multifaceted, they require many different approaches, not just going to the moon, which would be great also. We chose the term moonshot and used it because it captures 2 important things. It captures the ambition that we hope we'll see today, and we know we will, in the panels and in the conversations we all will together have with the panelists, and also the inspiration we wanted to convey. Besides the popular understanding of the moonshot idea, it's actually a little more complicated than that. At John F. Kennedy's speech in which he announced the moonshot idea, this is what he says. In a very real sense, it will not be one man going to the moon, it will be an entire nation. For all of us must work to put him there. I'd say her too, but he didn't say it at the time. That final point really captures the spirit of grand challenges. It's not a single researcher, a single discovery that is going to solve the challenges we'll be talking about today, but all of us working together for the benefit of humanity, broadly described. So, like everyone in this room, you voted with your feet today, and many times before. I deeply believe in advancing knowledge, in deepening our understanding, in sharing that knowledge with the world. I think that serves the greater good in so many ways. And it's not to get sappy in a room full of people thinking about the brain and neuroscience, but I think it's both a noble endeavor and an incredibly vital one. So I hope the things you take away from today include excitement about the work that is happening here and now at the university and all that is on the horizon for us. We are in a distinctive position to advance knowledge in ways that I firmly believe will improve the human condition for future generations. And myself, President Ryan, Ram, the deans of the schools, the department chairs, all the researchers, I think we're absolutely dedicated to this goal, and particularly the leadership is dedicated to the goal of this high ambition for the future. So thank you for being here today. Thanks for your spirit, your wisdom, the collaborative nature you've brought to this, and the expertise you're going to bring to today's conversations and future ones. I'm going to turn it over now to Megan Barnett, Vice Provost for Academic Initiatives, who's going to introduce the first panel and cover some of the details for today. Thanks so much for being here.
Good afternoon, and thank you all for coming today. As Provost Magill said, the faculty panelists have really put in a lot of work, and they've come up with some extremely exciting ideas that they're going to share with you today. But before we get to the fun part, we have just a couple of logistics. First of all, we are recording and live streaming this event. Secondly, as you know, we have 4 panels. They're each going to be about 45 minutes, and then we've set aside at least 20 minutes for Q&A and for discussion with all of you. And we really want it to be an interactive discussion and exchange of ideas. So during that portion, if you have a question or a comment, just raise your hand and someone will bring you a microphone. And once you have the wait until you have the microphone to start speaking. We have a lot of people watching on live stream today, and we want them to be able to listen to the discussion and hear the questions as well. We're also going to be taking questions, suggestions, and comments online. If you don't have a chance to ask a question live, you can go to the VPR's website, and there are Grand Challenges web pages where you can leave your comments and questions. So with that, we'd like to bring up the first panel, which is Cognitive and Developmental Neuroscience, Early Detection of Intellectual and Developmental Disabilities.
And it's my pleasure to serve as the moderator of this first session titled Early Identification of Intellectual Disabilities and Developmental Orders. I think best to just get started. So can I ask the panelists to introduce themselves? Dr. Kucenas.
Hi, I am Sarah Kucenas. I'm a professor of biology and the director of the program in Fundamental Neuroscience. I am fascinated by how the brain develops. And so I'm a developmental neuroscientist, and my lab uses zebrafish as a model organism, and we couple things like genetic manipulation, in vivo imaging, pharmacology, single nuclear RNA-seq to answer questions about how you build our favorite organ. I got my bachelor's degree at Valparaiso University, my PhD at St. Louis University, and I started here in 2009.
Thank you, Sarah. Dr. Cang.
Oh, this feels good. Hi, I'm J.C. Cang. I'm a professor in the Department of Biology and Department of Psychology. My lab studies the visual system, its function, its organization, and its development. Of particular, we use animal models. Of particular relevance to this panel is that we have been studying how sensory experience, visual experience in particular, shapes the connections in the visual cortex, among other visual areas. I guess one other thing I want to note is I received my PhD here at UVA 20 years ago, and I returned to UVA in 2017 after serving on the faculty of Northwestern University.
Thank you, JC. Dr. Beenhakker.
Good afternoon. My name is Mark Beenhakker. I'm an associate professor in the Pharmacology department. I received my bachelor's degree in neuroscience just down the road at Washington and Lee University. And then after that, I started a graduate program at the University of Pennsylvania, and that's where I really fell in love with neural circuits and the link between neural circuits and behavior. I studied that in the crab. As a postdoc, I moved to Stanford, and I carried my interest in neural circuit activity and behavior, but I started working on rodents. And that gradually evolved into an interest in the neural circuit basis of epilepsy, which is what my lab studies now here in the Pharmacology department.
Thank you, Mark. Dr. Solari.
Hi, my name is Emily Solari, and I'm a professor of education in the School of Education and Human Development. My work really looks at how we take basic science and fundamental science and translate that into practice around reading instruction and interventions. I'm particularly interested in how we identify children with reading disabilities early and how we intervene in a way that is aligned with scientific evidence.
Thank you, Emily. So, as a child neurologist, I often have to attend family meetings of a child being discharged from the hospital. This may be a child born prematurely, it may be a child born at term but suffered brain injury during the prenatal period or during the birthing process. It may be a child with congenital heart defects or a child with a genetic variant that's led to a brain malformation or neonatal seizures. And at some point during this family meeting, I start to feel all eyes looking at me, especially as we begin to talk about the neurodevelopmental expectations for this child. At that point, I often begin to talk way too fast. My hands start to wave a lot, and I start to just talk generically about the possibility of this or the probability of that. And as I talk, I can see the dissatisfaction on my colleagues' faces and the parents' faces, that they realize I've not really said anything specific in many ways to that child or given them a specific way forward for their child. And I would love to do better. I would love to be more specific. But I also recognize it's going to take a moonshot to get there, that we really need to begin to harness our understanding of nature and nurture to improve developmental outcomes if I'm ever going to be in the position to provide truly a personalized neurodevelopmental plan, one that I would hope we could even begin before that child's discharged from the hospital. So I think my question to all of you, but first Sarah, is do you see this moonshot as a possibility and one that we could achieve here at UVA?
Yes. As a developmental neuroscientist, I think it's a moonshot. Provost Magill, it suggests something quite ambitious but within reach. And I think here at UVA we have all of the parts necessary to kind of not only get to the moon, but to then bring this spaceship back home. How do we actually help these parents and these families, and how do we equip Emily and her colleagues, right, to make real change? And I think what UVA has is this deep, rich history of fundamental to applied neuroscience. And it bridges all across grounds, which there's all of us from all of the different schools here today. And I think we have the ability to really make transformative change so that clinicians and interventionists can help these families and these children, not only with early detection, but then with early intervention, so that we're kind of getting this double hit that allows for the most successful outcome for these kids. So I think what I want to talk about as a developmental neuroscientist, as I introduce someone who's just interested in how you build this organ, from my perspective, what needs to happen first. And so I imagine that, part of this moonshot is we have to get to the moon. We have to understand what this organ is that we're talking about. And so I wanted to kind of define, although I know I'm speaking to a room full of mostly experts, the problem, the magnitude of the issue, which is that the human brain has about 100 billion neurons. That's about the number of stars in the Milky Way. Okay, so that's a significant amount of cells. There are 10 times that number of glial cells. And so these glial cells are these non-neuronal cells that are considered support cells, but my and many of my colleagues in the audience know that these cells are actually the engineers of the nervous system. So 10 times the number of neurons of glia. And then these trillions of glia and billions of neurons have trillions of connections between them. And so it's this kind of community of interactions. And the number of connections, which is kind of astronomical, is actually more stars that are in the universe. And all of this is in the human brain. And we, from my perspective, don't know enough yet about how those connections are made. And so to create a system that allows for children to learn and to grow, we need to actually understand how the nervous system is formed. And it needs to form, and what's amazing about this organ is that it's hardwired enough for kind of basic life-sustaining things, right? Locomotion and survival and reproduction. But that it's plastic enough for learning and memory and repair and intervention, right? The nervous system is not a genetically hardwired organ. And so what's incredible, though, about it is that it has to develop, just like any organ system. And so the right cells have to be made in the right order, at the right time, in the right place. And then these cells have to then migrate huge distances to make partners with other neurons and glia. And then they have to interact. And then they compete, and some of these cells die, and some of them get replaced. And all of this has to occur on the magnitude of these trillions of cells. And that's all before any of these beautiful circuits that Mark and JC mentioned are even formed. And so how do we attack understanding that from the sense of kind of developmental disorders and early interventions? And I think part of the issue we have as a community is that we don't even know yet the level of diversity, the cells that are there, the systems that are in place, both genetically and environmentally, this nature versus nurture that builds this system. And so we have to, in my opinion, address this hurdle at some level when thinking about early detection and early intervention. Because we need to know who's there, what are the circuits, what are the interactions, because we have to know what's there before we can figure out how do we, right, genetically detect. Are there biomarkers? Are there clinical pharmacological interventions? Are there environmental interventions that we can create? But we first need to know who's in the room. And so I like to think about this kind of as like a car, right? When we all have check engine lights on, we go to the mechanic. But thankfully, the mechanic knows how an engine is built. That's part of the training, because to be able to fix the issue, right, you have to know how the thing is that you're trying to fix, the engine itself. And the goal, I think, right, is how can we make it so that either that check engine light comes on earlier, right, or that we don't even worry or wait for the check engine light, that there's actually other training and other signs that we can use before you'd ever get that kind of level of, right, we need to get to the mechanic, that there would be ways that potentially before discharge, right, Dr. Goodkin and others would have the tools to say, here's what we think this could look like, here's a plan. And so I think the groundwork has been set for this, both here at UVA and internationally. You know, a lot of the approaches in the last 25 years have been incredible. The first is, you know, kind of the creation of these connectomes. So we tried to approach how you build a brain by trying to look at the wiring itself, the circuits. And that's been incredibly powerful, right? This is work that has come out of places like Janelia and the Allen Institute, where we're mapping, quite literally, the connections in the brain. But the problem with that, when thinking about a neurodevelopmental disorder, is that's almost like a paper map. And some of you are the same age as I am, but we actually had to navigate the road on paper maps. It's not like Google Maps. It doesn't tell us what happened before the map was created or how those roads were created. And so it gives us a map, which is fantastic to understand the circuit itself, but it doesn't tell us how the circuit was formed, what genetics were involved, or was there a different plan before the road was created? Was there a reason why the mountain was cut through as opposed to not driven around? And so neuroscience, right, we have these connectomes, but then they also in the last 10 years have kind of realized that this isn't enough. And so there's been a huge investment in single cell sequencing, right? Okay, so we know the circuits, but now we need to know who's part of the circuits. And this is brilliant work, right? My own lab uses it, many of us do, where we're trying to identify who's there. And this is work that's just bearing fruit. Very recently, we've likely all seen these papers out of Science and Nature, these Allen brain atlases, where we're now getting snapshots of who's in the room. But those single cell data sets, as many of us appreciate, are kind of like IKEA parts lists. You open up the box and you know what's in the box, but it doesn't tell you how they got in the box or who they're really supposed to talk to or how they integrate into those circuits. And so I think the problem that still remains in developmental neuroscience that I think we at UVA are uniquely poised to address is now kind of adding in this biology and understanding of these maps and parts lists and creating an understanding of how nature and nurture, how genetics and environment drive these behaviors. And so what I think is incredible is that, like I started, we have fundamental to applied neuroscience. So we have people that are really interested in how you build these organ systems. And I imagine incredible partnerships with people with my lab, right, model organism people. And I think what's important is that the human brain is built with the same blocks and the same genes and the same cells as the mouse brain or the fly brain or the worm brain, right? The building blocks are the same. And here we have a huge history in developmental biology, developmental neuroscience, genomics, data science now, where we can literally have not just bench to bedside, but I would like to think zebrafish to patient, right? We know that there's a lot of resources coming out of some of these these GWAS studies. And what if we can find genetic signatures when we can, that we can then come back to my lab, to labs that use models, and say, here's this genetic signature, but we don't know what implication this has on the development of the circuitry. And then create models, quite literally, in labs that say, here are these, right, SNPs, here are these genetic signatures that we could then learn what this does to the circuitry, which would then allow us to understand more about what could be happening, right, during development. I think what's important, though, is that this doesn't need to be on a single cell basis. This is not that every patient would come from the lab. This idea is, what if we can identify signatures or themes that would allow us to understand major processes in development that if a child came through the NICU with some sort of genetic testing, we could say, well, here are these signatures. We now know from looking at our fundamental science that this is what can happen during development, or this is what happens to the circuits. And so I think that's fantastic. But I think what also we have, which a lot of institutions don't have, is we have the pipeline the other direction, where many of us are just fascinated with how you build a brain. And so there's fundamental discoveries occurring in our labs just for the sake of wanting to know how the system works. And we can then take that genetic information and then go back to our genomicists and clinicians and say, here are these genes we found that play important roles in cell migration, synaptogenesis, et cetera. Do we see these signatures or changes in these signatures in patients? And so I think having this pipeline back and forth here at UVA is something that's pretty unique that we don't find at many other institutions where neuroscience really is truly cross grounds. And it's not just me. I would like to say that there are so many people here, right? And I'm going to list a few because I just want you to get the magnitude across grounds. This is not an exhaustive list, right? But there are people like Jess Connolly, J.B. Morris, Adam Aribich, J.C. Cang, Sarah Segrist, Chris Detman, John Campbell, Kyle Lampe, Bettina Winkler, Eli Zunder, John Lukens, many people across grounds who are asking these questions about developmental neuroscience across schools. And so I think, from my perspective, this is totally achievable. We have to get to the moon first, and I see that first moonshot, this developmental neuroscience, is getting to the moon.
Thank you, Sarah. Thank you for elegantly defining the scope of the problem in terms of neural circuits and giving us an idea of how nature fits in there in terms of the genetics. So, JC, could I ask you to elaborate maybe more on the nurture side of a neural circuit development?
Absolutely. So thank you, Sarah, for making a really convincing, I think, strong case of the importance of fundamental science in this whole discussion. Like I said, like Howard said, I'm going to elaborate on the fact that one of the factors, nurture or the environment in its significance in brain development. As Sarah mentioned, our human brains are incredibly complex with billions of neurons, glial cells, and trillions of connections. These connections are also precise and selective. These are neurons are connected to form functional circuits to perform particular computations. But the level of selectivity and precision cannot be achieved by genetic factors or nature alone. Instead, in addition, it's profoundly shaped by the environment or nurture. I often use language as example, right? My wife and I are both Chinese. Our two daughters, of course, are Chinese by the genes they inherit from us. But they're growing up here and they speak English as their native language. What that means is brain circuits for language and speech are not hardwired, but instead are shaped by the environment. This brain's ability to adapt, to change in response to the environment, we use a jargon to call it brain plasticity or neuroplasticity, or the brain is plastic. But another aspect of this is the fact that I cannot speak English as fluently as my two daughters because I came to the States in my early 20s, suggests that brain plasticity declines with age. Right? So this intuitive notion of declining plasticity has been supported by numerous studies that demonstrated the existence of the so-called critical periods or sensitive periods in brain development when neural circuits are most sensitive to the environmental influence, such as sensory, motor, social, cognitive, and so on. Right? So these critical periods are obviously windows of opportunity, right? Because they allow us to adapt, not only to external environment, such as the language example, but as well as to individual differences. At the same time, these critical periods are also windows of vulnerability, because failures to receive proper experience during these critical periods are difficult, possibly even impossible to repair later in life. Right? For those reasons, this topic of critical periods have been widely studied in the field of developmental neuroscience, because it really gets to the heart of nature versus nurture, better yet, nature and nurture at the level of neurobiology. So one important question that we and others have asked is what makes the brain plastic, right? What turns on the critical period? More importantly, what closes critical period? If we know that, maybe we can use that information to rejuvenate the brain, to make the brain plastic again later in life. We have been studying this in the visual system. One discovery we made a number of years ago now is that by placing animals in an enriched environment, in this case mice, we were able to promote plasticity. Right? We have a clip there, I think. You can see that's not their typical cage, right? They are in a much larger space. There's enhanced sensory stimulation, motor activities, as well as larger cohort for social interactions. So, like I said, we showed that this was able to promote plasticity in both developing and adult animals. But later on, we actually figured out these mice, as you can see, they love running on the running wheels. Just by giving them those running wheels was sufficient, was enough to promote plasticity. But this is not to say that we can go out and run every day and come back be fluent in speaking a new language, right? This translation from animal studies to human work is much more complex. I think the key is really for us to figure out the molecular and circuit mechanism so that we can use that information for translation. Beyond this particular example, I want to sort of echo what Sarah just said. The moonshot, or the cutting edge of the field, is really at the level of a neurocircuits. For my field of visual neuroscience, most of our studies are still focusing on gross response magnitude, largely ignoring the precise circuits and the exquisite neural computations they perform. Right? But we know a purpose, at least one purpose of this critical period in the visual system is to wire up the circuits for depth perception. Right? Just take a step back. We have two eyes, yet we have one visual percept. And we can actually use these two flat images on our retinas to reconstruct the three-dimensional depth information of the visual world. Right? How does the brain do it? Our brain does it so effortlessly. Right? We still don't really know how the circuit is, what's the exact format of the circuit, and more importantly, how that circuit is wired up during development. Right? We do know it requires genetic cues, it requires sensory experience during a critical period for that to form, but we do not know exactly how. Right? That's where we're headed in my lab, hopefully revealing sort of general mechanisms for developmental neuroscience. But beyond that, as Sarah said, I think it's a good strategy for us to invest in this area of nature and nurture at the level of neurocircuits. Again, as Sarah mentioned, there's a lot of exciting neuroscience in the field that has mapped those circuits. Right? This BRAIN initiative has been funding efforts to map circuits in different brain areas, different species. There's no doubt that they will reveal more and more complex circuits, precise circuits. And the next obvious question is, how are those circuits established during development? Right? We at UVA, I think we are well positioned to tackle that question, to lead that direction. Right? So we have been world leaders in developmental biology and developmental psychology. And in between, there's many labs of developmental neuroscience, as Sarah listed, and many of which are actually working on brain plasticity during development. Right? So I'll list a few as well. There's Barry Condren in the biology department that uses fruit flies to study the circuits and the critical period plasticity for collaborative social behavior. Adama, who is speaking in the next panel, she uses mice to study visual system plasticity and preterm birth. And Dan Eliza is also in psychology, uses songbirds to study how sensory experience shapes the neural circuits for auditory processing and vocal learning. And Jess Connolly, of course, used both animal and human to study, in human, study how parenting behavior affects social development, social behavior development. Right? So all this is to say that we have real strengths in this area at UVA, and it's an important area, as Sarah just made a really strong case for, and it has great potential for improving human health. So it would be wise to invest.
Thank you, JC. So, Mark, we just heard from JC on how experience dependent processes occur in the brain and the concept of critical period. But could you take us from there to circuits and behavior? Once these circuits are formed, how do they lead us to talk and run and do all these things we need to do?
I can try to do that in a few minutes. Thanks, Howard. So I guess to begin, what exactly do we mean by a neural circuit? So as Sarah mentioned, the brain is composed of 100 billion neurons that are collectively connected by trillions of synapses. And those synapses are just the connections between two neurons where information is relayed from one neuron to another. And at those synapses, as Sarah and JC described, that's where you build the wiring diagram. The wiring diagram that is then shaped by environmental experience to ultimately produce a mature behavior. So in essence, you have a rough blueprint that is defined by genetics, and that is ultimately shaped by the environment. But the culmination of this combined biological building approach are hundreds of specialized neural circuits in the brain that drive all sorts of behaviors, ranging from the mundane but vitally important behaviors of breathing and regulation of blood pressure, all the way to the more fascinating but complex behaviors like fear, anxiety, joy, and grief. But to be clear, at the heart of each of those behaviors is a neural circuit that was formed during development. So if we want to understand human behavior, we have to understand the neural circuit. We have to understand how the neural circuits within specialized structures become wired up during development and how experience shapes them. And I think everyone here would agree with me when I say that in order to resolve this question fully, we have to understand how all cells, including those cells that are not neurons that Sarah mentioned, the glial cells, contribute to this neural circuit basis of behavior. It truly takes a biological village for a circuit to produce a behavior. Where was I going? Oh yeah, so non-neuronal cells. For example, non-neuronal cells such as microglia are instrumental in shaping the wiring diagram during development. And we are fortunate to have someone like Ao, who is a world's expert in microglia function here at UVA. Another non-neuronal cell, known as the astrocyte, is instrumental for shaping the electrical signals that are produced by neurons. And with the recent arrival of Harry Sonheimer, we now have one of the world's experts on astrocytes. So we have the relevant expertise to begin to understand how that diverse biological village enables a neural circuit to produce behavior. So how do we resolve neural circuits? This gets back to the point that JC made. Through the BRAIN Initiative, the NIH and the NSF have provided significant support for the development of tools to both monitor neural circuits and to interrogate neural circuits. And these tools have truly revolutionized neuroscience research in the last 15 years. We now have tools where we can control neural circuit activity with light through a tool known as optogenetics. And this is actually one of my favorite examples of optogenetics, provided by our very own Steve Abbott and Patrice Guillenet in the Pharmacology department, where with light, they can shut off the neural circuits that control breathing. So when you see that light turning on, the animal stops breathing. Another approach that we have that has recently been developed in the last 15 years is being able to monitor, to record the activity of large populations of cells as they generate these neural activity patterns. That is the neural code that we have to resolve, the code that the circuit produces that generates behaviors. And with these tools like calcium imaging or optogenetics, the neuroscience community is beginning to understand the neural circuit basis of behavior. And these tools are being used by UVA scientists every day. So as I mentioned, Steve Abbott, Patrice Guillenet, Doug Bayless use these tools to understand the neural circuits that drive breathing. Adama and JC use these tools to understand how experience shapes the neural circuits that control vision. Jaideep Kapoor and Manoj Patel use these tools to understand the neural circuits that drive epilepsy. Ali Guler, Mike Scott use these tools to understand metabolic disorders and addiction. So I think by bringing together these experts in neural circuits, together with the experts that we have at UVA that study development, that JC mentioned, and together with the experts that live over in the world-class brain immunology and glia center, or big center, I think that we can collectively begin to understand what I think is a truly fundamental question in neuroscience. And that is, how does that rough blueprint that is shaped or defined by genetics, one that is shaped by experience, how does that ultimately produce a healthy brain that is capable of controlling breathing, that is capable of learning new languages, that is capable of feeling complex emotions? And more importantly, or as importantly, I would say, how does disruption of that combined building approach lead to developmental disorders? So I guess what I'm saying is that I think we need to bring scientists together. And this is something that I heard recently. Philip Sharp, Nobel Prize winning scientist at MIT, recently visited UVA, and he was asked, how do you spark discovery? And his answer was clear: bring scientists together. Let them work closely together. Promote their dialogue, promote their conversation. That's how you provide the substrate to spark discovery. I think another way to spark that discovery is to grow the community, invest in new faculty. New faculty bring new ideas, new faculty bring new tools, and those ideas and tools then percolate into the existing neuroscience research community to make all of our science better, okay? And actually I was thinking last night, Howard, how could we support this? And my idea that we may want to consider is maybe we can levy a heavy tax on the super wealthy faculty at UVA to support this idea. Maybe the 70 wealthiest faculty at UVA. Just an idea. You're looking a little nervous, Howard. Yeah. Okay, so in the last few minutes, I just want to bring it back to sort of the concrete ideas of our moonshot. So the basic science portion of our moonshot is to, as Sarah described, provide snapshots of neural circuit development throughout lifetime. And to understand how genetic programs and molecular cues generate those snapshots and how experience can shape those snapshots. And our panel thinks it's vitally important to do so both for the healthy brain and for the distressed brain. Ultimately, Howard, our moonshot hopes to provide information that gets at how the neural circuits in the patient you described, how their trajectory is affected by that early life trauma. But we believe that to understand that, we have to know both how the healthy circuits develop as well as the distressed circuits develop. We need to know both to fully resolve that question. And the last thing I'm going to say, importantly, these trajectory-changing interventions that we envision do not necessarily rely on pharmacological tools or surgical intervention. In fact, we think that just by altering experience, we might improve the trajectory of patients. And for this reason, we have Emily on our panel. So Emily is from the School of Education and Human Development, and we hope that by harnessing the knowledge that the basic scientists provide to understand how nature and nurture drive the development of neural circuits, that Emily can ultimately use this information to provide real, tangible boots on the ground for change to improve the trajectories for children. In essence, as Sarah described, we hope that Emily will be able to navigate our moonshot rocket back to UVA, back to the Charlottesville community to implement real change.
Thank you, Mark. career in politics, thinking outside the box. But I think we all agree, and I think this room shows that we realize that none of this work is going to happen in isolation, that it's going to take clinicians like those in my department in developmental pediatrics, the neonatologists, teaming with the scientists, the educators, and everybody else if we're going to be able to harness nature and nurture. So Emily, Mark got us to the moon. We don't want to leave any space junk on the moon. We need to navigate back to Earth. So can you get us back down here?
Well, I can certainly try. I want to thank my colleagues for tasking me with the pretty small job of getting us back to Earth. When we talked about this earlier, one of the things we talked about is that getting to the moon and getting back to Earth for implementation in real-world settings requires a different skill set, but it also requires communication between the two sets of teams. And so I'm going to talk a little bit about translational science and what that looks like through team science. But the basic question here is, how do we use fundamental science or basic science to inform what's actually happening for kids in communities, in schools, in their homes? Because that's the goal of basic science, right? I think it's to translate into what's happening for our community. And so really when I was thinking about how do I tackle this very small topic, I was thinking about JC's works and Mark's works and Sarah's work in fundamental science, and how do you connect that to kids in homes, communities, and schools, and how do we have this sort of really intentional collaboration between basic science and applied science that makes sense? I would suggest that translational science, when done in teams with applied scientists and fundamental scientists and basic scientists, is perhaps slower science. And we need to consider that, and we need to be okay with that. One of the, we could all go back to our own labs to continue doing our own science in our own silos, but I'm not convinced that's the way that we can make change for kids on the ground and for families and parents who have children with developmental disabilities. So we know that there's a developmental science literature that demonstrates that early environment is really very important for the development of children, both typical and atypically developing children. And this is true for their development from childhood through adolescence and into adulthood. And we also know from my colleagues that the basic science is there as well, that there are certain parts of brain development that are essential for healthy development, and certain parts of brain development that things happen, and that sometimes results in a developmental disability. I would suggest that both of these bodies of evidence are essential, and they need to talk to each other. We need to be talking to each other in order to make real change for kids. I'm going to talk a little bit about reading, because that's actually what I know about. And when we think about reading development for kids, especially kids who are at early risk or who come from subgroups of kids who typically have risk trajectories, a fundamental question that we ask is, what is the basis for responsiveness to intervention? Why do some kids respond to our intervention or our environmental inputs, and why do some not respond? We know that in reading, there are critical periods, as JC suggested, wherein if you are able to identify reading disabilities earlier and remediate them or treat them, kids typically do better longitudinally. The NIH in the early 2000s did a series of studies in this and showed that if you wait until 4th grade to intervene with a reading disability, it takes 4 times as long to remediate that issue as it does as if you were to do it with kids who are in 1st grade. So this really drives home the importance of early identification of reading disabilities. We also know that reading disabilities, just like other developmental disabilities, are influenced by genetics. There have been decades of studies to show that, through twin studies showing the influence of genetics on reading development. But we also know that genetics are not the end-all be-all. So the genetics does not 100% predict, how well you're going to do in reading over a lifetime. And we need to think about what are the evidence-based interventions or environmental inputs that can impact trajectories. And there has also been some research done to show that when you implement evidence-based interventions for kids with a reading disability, that the brain structure does reorganize a bit, and it starts to look more like a quote-unquote normal brain or a child who does not have a reading disability. And that's really important to know because that can inform our practice. But I would suggest that that might be the easier science, and my colleagues may not agree with me. But knowing how to do intervention related to any sort of developmental disability, and knowing that it does change sort of the way in which the brain is functioning when it encounters words on page is one thing. But knowing how to implement that in a community, in a school, in a home is a whole nother thing. And the way how we have to think about how we're translating our science. So my challenge is, my research is not the basic science and the fundamental science. It's taking what we know from that space and translating it into real world practice. And this is in early childhood context, in elementary schools, and in community contexts. So talking a little bit more about translational science, Howard started this by saying he often has meetings with parents, and parents want to know what to do tomorrow with their children. That is the same thing I hear weekly from teachers in our schools, from early childhood care providers. They want to know what to do tomorrow. And so my burden is to take what we know from science and translate that to better inform them on what to do. I think in order to do this translational science, as many of my colleagues have suggested, we need multidisciplinary teams of people who are intentionally coming together to talk about these issues. I will also pause just to say that in the planning of this panel, I learned so much from my colleagues that made me think really critically about the work that I'm doing in communities. And I think that's back and forth. So fundamental science can inform applied science, and applied science should be informing the fundamental science. I think we need to think about how we're communicating our science broadly. Every translational science team should have someone who's really excellent at communicating science. Because I think a lot of us in this room would agree that researchers are actually terrible at communicating science. A lot of us are. And what is the point of our science if it's not impacting the lives of children? That's my sort of area of research, children and families. And then the other thing we need to think about, many of us in this room also work with clinicians and practitioners. I work to think about how we best train teachers around reading. And so once we've established evidence-based practice, how do we then train our practitioners to implement those practices? They have to have a deep knowledge of the science, they have to have a background knowledge of the science that is deep enough for them to use that in their everyday practice. And I think this gap between research and practice is large. It's certainly large in my particular area of reading. If you think about reading development and reading disabilities, reading is perhaps one of the most studied aspects of human learning. But we just aren't doing a great job teaching kids how to read in our schools. So there's a big gap between what we know to work and what's actually happening in the community settings. And then lastly, I think we need to think about how we're engaging our broader community and getting feedback from people on the ground. I like that Mark thinks that I'm the person on the ground, but also I do work with folks on the ground, but I'm also here in the university, and so we need to be engaging with our community members around what needs do they have, and how can the science that we're developing here inform their practice. I think the ultimate goal of scientific research is translating basic research findings into products, processes, and policies that inform and improve the life for our broader community, and I think that should be our overarching goal. I'll stop there.
Thank you, Emily. I'm not even going to try to repeat what you just said so elegantly about the need to make sure that what we learn here gets out. to other places in our community. And that brings in a whole other panel, obviously, of how do we do that, and the social determinants, which allows some of our communities to do well and others not as well as we would hope. So, Sarah, you led us off on this mission of harnessing nature and nurture to improve developmental trajectories. Guess I'm asking you to summarize everything we heard today in a very short amount of time.
Okay. So, I mean, I think what I heard, right, is we have this moonshot, right, where we want to have earlier detection and earlier intervention, right? But those are kind of what you heard from us, 2 halves of the trip to the moon. I really think that there's 2 different crews, right, that need 2 sets of astronauts. And with space shuttles, there's often the launch crew and then the landing crew. And so I think what we heard is to get to the moon, the first half of the moonshot is this role and understanding of genetics, right, nature and nurture, genetics and the environment, in developing these systems, right? Who's there, how are the circuits wired, and where are they plastic? And having that fundamental understanding of how the brain is wired will then allow Emily, apparently on a solo trip, no, but to come back, to take that fundamental information, right, and understand where these critical periods are and where the genetics is a little bit more wiggly than we thought, right? Neuroscience is not as hardwired as previously appreciated. And so these 2 shots, right, getting to the moon, understanding the circuitry, understanding the role of nature and nurture, and then integrating that in a real world community way that allows Emily and the teachers and the parents and NICU staff and, right, pediatrics to then consult with families and teachers so that these kids are leaving with an idea, and not the kids themselves, their families, but they're leaving with a plan, or at least a better understanding of what, right, they could potentially be doing to enhance, right, the trajectories of these kids.
Thank you, Sarah. JC, Mark, Emily, anything to add before we open up to discussion?
I guess just one thing that was sort of mentioned by Mark already. I really echo that we need new blood, we need new crews in this endeavor by hiring new faculty at the early stages of the career so that can bring new energy, new excitement, new tools to our community.
Emily.
I would like to suggest that I'm not on a solo trip, and I failed to mention that there are many other folks across the university who do this research, thinking about how we best implement practice in early childhood settings. Certainly a lot of folks in the School of Ed, people who do prevention science, so how to sort of prevent difficulties before they happen within the context of schools and communities, and there are other people around who do similar work.
So I guess I'm going to open it up to the audience at this point. I just would ask that you introduce yourself for those who are at home listening. Raise your hands, anybody?
Hi, I'm Nadia Lunardi. I'm an associate professor of anesthesiology. So I'm a clinician and basic scientist and early stage investigator. In this moonshot theory, which I like and I've heard a lot of interesting and exciting things about it, how do you see implementation of a strategy where younger folks like me have access to the collaborations that you're talking about, and maybe some of the funding that will be available, so that by growing the local community, that becomes also helping younger folks like me to get to the stage of all of you?
Thank you, Nadia. So let me summarize for those who may not have understood through your mask and make sure that I understood. The question is, how do our junior faculty with an interest in this area also get to be involved in our teams and grow and help get this rocket to the moon and back?
I'll start. I guess I'll jump in and I'll say this is, I think, an answer, but hopefully, one of the reasons that brought me to UVA many years ago was the collaborative, open, interactive neuroscience community. And I think our panel, at least, and I would imagine all of the panels, because we all know everybody here, is that, you know, these are not exclusionary projects, right? This is an idea where we would want everybody who's excited and interested, and we would welcome anybody, because this is a big moonshot. I think we overshot the moon, right? I think, you know, we need the expertise of people, and I think what UVA has, again, we keep using this word, but many of us have been many places. It's pretty unique here. The community here is open, and we're collaborative, and we're across grounds, and, you know, we're just simply representing one field that we think would be amazing, and so we would love anybody, but particular junior faculty who want to contribute and help get this project rolling.
Well, I would consider ourselves still junior faculty as well. I'm joking. No, like I said, I think junior faculty should be the key driving force of the whole process. By utilizing the skill sets and expertise of our existing junior faculty, and also by hiring new junior faculty to join this effort. So I think that's really the key.
I saw another question pop up.
Hi, I'm Rob Hewitt. I'm no faculty affiliation, but I remember reading an article, I think it was in the Atlantic a couple years ago, about a building at MIT that was built during World War II, and it was sort of a nondescript building that people could come in and tear up and fit their labs in and so forth. But it created an interesting group of people, people from like linguists like Chomsky to physicists and other people, but just casual conversations in the hallway that instigated really creative ideas. So I'm curious, I'm really excited to see you all working together, but I'm curious, how often do you have the opportunity to have these sort of in the hallway conversations that kind of spark that creative idea?
Not often enough.
I totally agree. I wish we could have them more often. I really, really do. It's crazy. So there's on a smaller level, this happens with graduate students all the time, where, you know, you're having a conversation with your graduate student about their project, and just through the circumstances of that conversation, who knows what it is, but you finally you have that spark that drives the project forward. And that always, you know, that drives graduate students crazy, because they are convinced that you're withholding that information, right? But the way I like to look at it is that, you know, let's say you have 10 conversations, right? Maybe one of those is spark producing, right? And who knows how it's spark producing, but it is. So if you can have those 10 conversations across a week, as opposed to 6 months, I think that's how you really spark discovery quickly. And as JC mentioned, I mean, I hang out with JC once a week because I go to his journal club, but I wish I could hang out with him every day. He probably doesn't feel the same way, but you need to. It's so hard to understand the circumstances that drive that spark, but I think for sure you just have to promote its occurrence.
Yeah, I would add that during COVID, we've had to be much more deliberate about having those conversations. But even before COVID, I'm very fortunate to be in a very rich epilepsy science community here, where clinicians and epilepsy scientists have been brought together, thanks to work by the Brain Institute, fostering conversations about sudden unexplained death in epilepsy. And so bringing those clinicians together with the scientists is quite fruitful, but one does have to be deliberate about making sure that that happens.
Hi, Jennifer West. I'm Dean of Engineering, and this looks like kind of a very broad, somewhat vague moonshot right now. And so if we think about wanting to have early detection of kind of any kind of intellectual disability early in infancy or childhood, what is it we need? Do we need kind of to identify more genetic profiles associated with disease? Do we need new imaging technologies? Do we need new ways of doing behavioral screens? Like, what is it that needs to happen to actually enable this moonshot?
I think all of those things, right? I think the idea that we'll be able to diagnose somebody the moment they're born with a genetic test as being 100% possible of having dyslexia, right? That's not true, and I think that's because nature and nurture are involved. But I would imagine that genetics can go part of the way by saying maybe if there's family history or if there are circumstances during pregnancy that might predispose a child, that we could potentially, right, have genetic markers, if you will, that would allow clinicians to kind of have an idea, and then by using fundamental science to understand what those might do to imaging. What does that do to circuit activity? And in a mouse, maybe this genetic signature causes a delay in circuit formation, and we might say to that family, we would love to see your child at 6 months. We're not going to wait until they're not reading in kindergarten, right? And maybe using that fundamental science to say, let's create a plan where at release, we have an idea of what could be the future. Now let's follow up before we have any need for the parents to come back and say the child isn't reading, or the child isn't responding, or the child, right, is not maturing as fast as a sibling. That's how I imagine it, is that it's additional sets of tools that allow us to earmark patients and families and say, here's what we think is going to help, and if we hit any of these points where the imaging looks slightly different than what we expect, then we're going to need to talk about an early intervention plan where we know that this critical period or this plasticity or this help that could come from these interventionists might actually change the trajectory of this outcome. What do you guys think?
Yeah, I mean, so imagine, this hypothetical scenario where you have a mutation in gene X, and once you've landed on the moon and this moonshot has been realized, you can understand how that mutation alters a neural circuit so that it's different at year 3. And then imagine if you knew how that circuit was altered and how you could then use environmental enrichment, for example, to tweak that circuit to improve it. Like, that is one example by which I think you could, as Sarah was mentioned, sort of predict the developmental trajectory and then use specific enrichment schemes to benefit that circuit so that it becomes normalized.
I may also add, I think we really just don't know enough about the basic process already. I can use one particular example in my own small field, which is about critical period in the visual system. In that study I cited, this was 5-6 years ago, we showed actually the timing of the critical period is critical. Having it open too early is not a good thing. Now there's emerging evidence in the field showing having it open for too long is not necessarily a good thing as well. I think that in part is from others' work and also work from one of my former postdocs. I think we're still at really an early stage of understanding, for example, brain plasticity. We just don't know enough yet.
Left. Okay.
This is just Liz Magill. This is a quick follow-up to that question, Jennifer's question, which is, Emily, I know that you have an army of people who do some of the work that you do, right? You said at the end that there are others, and can just, especially for the basic scientists, the developmental neuroscientists, can you talk about all the different translational sites where you could assess and test detection and intervention? I think you have access to lots of classrooms across the state where this could be translated.
Sure. I mean, we did have a lot of access pre-pandemic, and I'm hoping that we have a lot of access soon. But I think, is the question, how do you move this into community settings? One thing I also failed to mention is that very recently, this past year, the School of Education hired a new faculty member who does neuroscience and education work. And a lot of her work, she will not be here until January 1, but a lot of her, she does sort of this groundbreaking work sort of looking at understanding children's experiences within classroom settings and how that's impacting their development. And I think, there's multiple ways that we as education researchers screen for risk, and usually that's behaviorally, right? That's through assessments and screeners. Is there a point where we could be in schools doing some of this more, is other ways to identify risk? Potentially. Are we there yet? I don't think so, although my colleagues may tell me differently. But I think there is an evolving science around neuroscience and education specifically, and how can we use that? You know, in my role in that is how do we use what we find from those studies to develop and implement precise interventions. So this sort of like precision medicine, but really it's around educational interventions. I don't think we're quite there. I think a part of this would be sort of moving that way. I don't know if that's...
Fred or Harry?
Harry Sondheimer, I'm in neuroscience. You did a beautiful job, developing this theme, from cells and nurture nature to circuits. There's seemingly a larger gap going to Emily and going to, learning disabilities at the level of reading and whatnot. And there's a lot of risk in the development of a child that's actually in the womb still, embryo, and in a neonate. Have you given thought to how you could insert someone between Mark and Emily to fill that gap and make that more of a continuum?
Yeah, it's a great idea.
Hi, Kyle Lampy. I'm in chemical engineering. I really appreciate the analogy of the moonshot in terms of aiming big, but I might be a bit provocative and say that that's not aiming big enough, because we've been to the moon before, not all of us, but we have the technology to do that, right? And maybe Mars is dreaming a little bit bigger, but we have the technology probably to do that too. We just don't have the life support system to make it happen. And as an engineer, often described as a tool developer, although Sarah called me a developmental neuroscientist today, I will have you know. We know what we know, and then there's the whole we know what we don't know. But if we say instead of going to the moon or Mars, we want to go to Pluto or the next galaxy, what kind of tools, the we don't know what we don't know bit, and how do we take a bridge to get to that point? What tools maybe, how do we discover the tools that we might need?
The way I see it is having everybody in the room, right? I mean, I always like to say that everybody's a neuroscientist, you just don't know it yet. And I think part is having, right, Chem E in the room and having every school, having everybody who's got an interest together talking, right? Because I come from a very specific, you heard that, I'm very different from JC, who's different from Mark, but we all have this expertise, and being able to be in the room and talk about these things, this close proximity, right? This ability to have these hallway talks and our trainees to have these interactions, that's how we learn what we don't know, right? What we don't know yet.
I totally agree with that. Maybe one specific example to the unknown unknowns. Mark mentioned a lot of beautiful tools that's working really well in mapping out circuits. Many of those tools do not yet work well in young animals, in young beings. So that would be, I think, a brilliant direction to go.
Or in glia. Those trillion cells, these neural tools don't work well in those cells at all.
I think an additional component that we didn't describe is our computational resources, like building the computational models so that we can, at least in theory, construct these circuits to give us a glimpse of how they might work. I think that's always going to be an important component to all neural circuit science. And one, of course, that dovetails really well with the expertise that's also being grown here at UVA.
I think I saw Jadip had the microphone, or Kevin had the microphone.
I'm Kevin Pelfrey, developmental cognitive neuroscientist, and this is just to resonate to Dean West's question and also to Harold's comments. I think that intermediate step that's kind of missing is a real understanding of trajectories of human brain development early, using tools that we have readily available. You know, we're scanning babies with fMRI, we're beginning to do fetal scanning, we're doing early scanning with EEG and optical imaging, and really understanding those trajectories. And we'll talk about this in the context of autism. But putting that data together with data about whole genome sequencing, about immune function, the microbiome, and bringing that together in the context of a data science community that can implement the types of methods we need to get markers that aren't snapshots, but rather are trajectories over time that are multimodal and inherently multidisciplinary, then those can become the targets that those who study animal models can actually begin to try to unravel, as opposed to targeting, you know, an autistic mouse or an intellectually disabled mouse or a mouse that has seizures, but rather the target of brain development that differentiates those three different types of intellectual disability, which we know share common genetic roots with each other, with schizophrenia, with ADHD, with every other, with typical development as well. So just wanted to put in a plug there for developmental trajectories in humans.
Thank you, Kevin. I think moving from scalars to vectors is important. I think Jadip did have a question.
No, I was just going to respond to the invest. I think we need to invest in real terms, developmental neuropsychologists, developmental pediatricians, developmental psychologists who can do the kind of work that Kevin just said. We just don't have enough people doing that work that Kevin just put out and they're proposing. So going between JC and Emily, we need a bunch of recruitments that would actually sit between, so that Emily is not bringing the whole thing home. She can't. No one person can. She needs help, and help means more recruitments in these specific areas of imaging and neuropsychology and psychology at the interfaces between these things. So we should be obvious where we are going. We do need more people to do this.
Question in the back.
Oh, sorry. Hi, I'm Aminata, and I'm a postdoctoral fellow in the neurology department. This has been really amazing for me as a trainee to see how all of these big shots are thinking about the future. One question that I have, I think that one thing that's clear from what you've all expressed is this idea of understanding how to make a neurodivergent brain more neurotypical. But is anyone on campus or has anyone thought of the idea of maybe trying to understand the potential of a non-typical brain development and how we can devise strategies to make those type of brain more, not to fit in the norm, but to make a leap to a better world, I guess.
Can I try to repeat the question?
Yeah.
It's hard to hear up here, so if we look puzzled, it's because it takes us a minute to make sure. So the question was, I think, please correct me if I'm wrong, right, we've been talking quite a bit about typical brain development, and the question is there work at UVA, or are we considering how to make atypical brain development not typical, but better tuned for outcome? Is that correct? Okay. I repeated the question. I mean, that's a fantastic question.
It's a great question. I think Kevin can probably be better.
Yes.
On the next panel, there's a lot of experts on the faculty that's working in that area. But for our panel, I guess I will try to make a case that you really have to understand how typical, in quotation, brain development occurs in order to understand how to improve the trajectory of atypical development. Right? You have to know how clock works before you can fix it. You know, that's like auto mechanics, like what Sarah mentioned in her analogy.
Oh, we're done? Okay. Okay. Fred, you get the last question.
Yeah, great. Really interesting discussion. I wanted to come back to the point that Jennifer West made and others followed up on. And my question is are there some prototypical examples that you can think of where fundamental biologists have discovered something unique in the lab and that has been translated, to enhance learning outcomes or some other things? So is this a moonshot where really no one's ever gone before? Or are there a couple of examples where, we've seen translation of a fundamental discovery to more of a practical implementation?
Do you have any examples?
Yeah, I can.
Go ahead.
Sorry, I can start with a very basic. I hope that answers that's the example that you're asking, right? Again, the system that I use to study the visual system plasticity, they started back in the 1960s from David Hugo and Torsten Wiesel. And they showed if you suture 1 to 2 eye strut in the developing kittens, that's going to result in amblyopia and strabismus in the animal models. That led to this realization we really need to correct that brain deficit, that deficit in visual experience as early as you can. I think that led to changes in practice in ophthalmology. That would be one example. But again, we don't really know the exact developmental process during that visual preparation process.
I would suggest that reading is one of them. And I think importantly though, we know a lot about what's happening when kids are learning how to read and why some children struggle to learn how to read and write. And I guess I want to go back just really briefly to just because we know that doesn't mean having that knowledge is changing trajectories for kids in schools and communities. And so that translational science piece, I think, is essential because we have to be able to take what we know and use it for better outcomes for kids. And there is a breakdown. And a part of that is communicating our science and engaging our communities and understanding that schools and communities are broad and complex. We are working with human beings. There's heterogeneity within every single teacher that we work with, every community leader, and that is a really difficult thing to tackle, how you translate the science into real-world settings.
Thank you. Thank you, Emily. I just want to thank the 4 of you. This has been a fast, it's been a busy, but it's been a fun process. And I've enjoyed generating this idea with you and watching where we went on this journey, so thank you.
Thank you.